All right.
All right. I think
So,
that we have enough students, so
yeah. So I
let's
try to upload. It's not coming up. Maybe I have to, now it's there.
Yeah, it is. Okay, so
Okay.
you can start whenever you want. And thank you very much, Marcus.
Yeah, thank you. All right, so I start the second part of this Erasmus program on the virus side, and that's the role of viruses for microbial diversity and food web dynamics, biogeochemistry, and in a final slide, climate change. So here I introduce a very important concept in viruses, that's the viral shunt, and do again a little bit of historical things. That's that's the grazing food chain, that's how it developed in the ocean and also in other parts like other aquatic systems. So we have primary producers in the ocean, that's phytoplankton, and these are eaten by grazers, and the grazers are eaten by carnivores. The primary producers take up the inorganic nutrients, and then the grazers eat. The primary producers, they produce also inorganic nutrients. The grazers here would be the protists. So that's called the grazing food chains. Then in the 70s, it was realized that primary producers produce not only phytoplankton biomass, but they also release a lot of dissolved organic matter. Extracellular release, that's called. You will hear that from other scientists, I guess, during this course. But also when grazers eat primary producers or carnivores eat other grazers, they produce also dissolved organic matter that's called sloppy feeding. And this material would be lost from the food web if not heterotrophic bacteria wouldn't take up this material. And heterotrophic prokaryotes are also eaten by the grazers, by the protists. And that's called the microbial loop concept. And then in the 1990s, or end of the 1990s, there was another concept developed that's called the viral shunt. That means that all these organisms, primary producers, grazers, carnivores, and especially heterotrophic prokaryotes, are lysed, infected and lysed by viruses. And what's happening here is what you see on the lower left side is that viruses come out of the cell. They leave behind either this bag, as you see here, or the cell wall disrupts completely. But also, when the cell opens or disrupts, the cell content is released. And this produces dissolved organic matter. I come back later to that with some numbers, and that's called the viral shunt. Okay, and this is a picture of that comes from a Nature paper. There you see the viruses coming out, and in green you have all the ribosomes, and the cell content is getting out of the cell, and this can be used by other bacteria. That's a very important factor in regeneration of nutrients and organic matter. That's another way. You have on top to the right, you have a cell where the viruses are coming out, and you see that the cell is already disintegrating. And this material is then used by other bacteria. And the viral shunt here shown in green is, when you have a cold and you sneeze and all the things, you also produce organic matter, and that's also happening with animals, with zooplankton. The protists will, because they are single cellular, the cell will burst, as well as phytoplankton and the prokaryotes, and that produces this DOM, that's called dissolved organic matter, or colloids are produced, or particulate organic matter. You have probably, I don't know whether you have already introduced into this concept, but dissolved organic matter and particulate organic matter usually defined by the type of filter you're using. So you remove, that's usually nominal pore size of 0.45 micrometer, and everything that's smaller, it's called dissolved organic matter, and everything that's bigger, it's called particulate organic matter. The color, it's a sort of a in-between material. All right. It's a special environment here. I discussed the deep sea, and the deep sea is mainly fueled by sinking particles. These sinking particles come from decaying phytoplankton, for example, or by fecal pellets from zooplankton, or as I said also yesterday, that black carbon or soot particles or Sahara dust or volcano ash particles take up a lot of organic material, and this sinks down, and this fuels mainly the dark ocean. The dark ocean is starting from the mesobiologic area, like in about 200 meters. There is some in situ production from hot vents, but quantitatively most comes from the sinking particles. That means detritus and then DOM that's distributed or dispersed from this detritus. And then what you have in the deep sea is in the mesopelagic layer that goes down to 1,000 meters. You have the prokaryotes taking up this organic matter. They are eaten by protists and they are eaten by zooplankton. You don't have very much zooplankton below 1,000 meters. But in the sediments, you have so-called meiofauna. It's a nematode. They go down to, down to the seafloor. And then you have, of course, the viral shunt. And this is a food web with reduced complexity that is fueled by the sinking particle. But you still have this viral impact down there. Okay, now, how do viruses influence prokaryotic diversity? Prokaryotes, again, are bacteria and archaea. So we have this concept that lytic viruses, if you remember from yesterday, the killing the winner concept. So the viruses keep in check competitive dominance, and thus allow for the coexistence of prokaryotic species. That's one of the ways. But what I just talked about a few minutes ago, there's this release of the so-called lysis products. That is when a cell lyses and releases the cell content, and the cell walls also usually broken in several parts, and this can be taken up by bacteria. This material can be taken up by prokaryotes in different ways, and they can also influence prokaryotic diversity. We have also two other possibilities. Yesterday I talked to you about lysogeny, and lysogeny means that the phage genome is integrated in the host genome. It's then a sort of dormant stage, but it's not totally dormant. It can, for example, protect the host against similar viruses, and it can also confer morphological and metabolic traits to the host. For example, it can change the cell surface of the cell so that it cannot be eaten by protists anymore. And then from the virus side, there is also a mechanism that's called transduction. That is when viruses by error take up some host DNA and transfer it to the next host. And the next host can then take it up. And if this is a useful material, this is potentially incorporated at a low rate, but possibly into the host, and this can mean that the host can acquire morphological and metabolic traits. So these viral genes and viral activity increase prokaryotic diversity, and this is of course then a target of selection and a source of ecological functions. So again, a little summary of that, what I just said now. We have the lysis and the killing the winner idea to do the consequences to keep in check competitive dominance, and this has been clearly shown in a lot of experiments with natural communities. The release of lysis products and the influence on prokaryotic diversity and production has been shown in a lot of isolates. There's also another interesting point, because when the viruses lyse the host cell, they'll produce enzymes to destroy the cell from inside. But then these so-called phage-borne enzymes act differently on the organic matter once they are released, and this has also an influence the processing of the organic matter. And then in lysogeny we have the so-called phage conversion, which transfers immunity and phage-encoded morphologically metabolic traits. That has been shown with isolates, and transductions has been shown with isolates and genomics or metagenomics. Okay, the killing the winner hypothesis. That I will explain now a little bit. The killing the winner hypothesis is a simple model and method or model to explain the different types of strategies you have for prokaryotes in the ocean. It's of course simplified, but it's a bit more complicated than most models, because most models only think about what bacteria can take up, that is free resources, which means nutrients and organic matter. But this model distinguishes between two different groups, and the one is the competition specialist, which means these bacteria can take up organic matter and nutrients very well. So this is the so-called winner. That's why it's called killing the winner. And they're killed by predators or parasites, because they're good in taking up these free resources, but they are not good in defending themselves against predators and parasites. And then you have another group of bacteria that's called the defense specialists. They are not so good in taking up the free resources, but they are very good in defending themselves against predators, that is protists or parasites, that is the viruses. And this is also to explain the so-called Hutchinson's paradox of the plankton, which means why are there so many species in the ocean, although the limited amount of the amount of nutrients are extremely limited. Okay, I'll skip that one. Here we have the way how bacteria can get genetic information from other bacteria or from other sources, and there are basically 4 types, and that's called the horizontal or lateral gene transfer. The vertical gene transfer would be when cells just divide. Okay, so the one is conjugation, when 2 bacteria join and exchange DNA. Then you have transformation, where bacteria take up free DNA, that's like a part of dissolved organic matter, and they can use that in their own genome if there's something useful for them. Then we have transduction. That's what I mentioned already, and that means that viruses sometimes do not pack virus DNA into their capsid, but they pack in an error host DNA into their capsid, and they infect, and this material can then potentially be used by the new host cells. And then there's also another thing that's called the gene transfer agents, and usually the gene transfer agents are viruses or bacteriophage that are not functional anymore, but that in the way that they can produce new viruses, but they still can transfer genetic material. Okay, then we did an estimation for using data from the Mediterranean Sea, like the diffraction of lysogenic cells, what we talked about yesterday with FLCs, prokaryotic production, burst size, estimations of induction frequencies, and a conservative number of transductants per infective bacteriophages in seawater, and the volume of the Mediterranean Sea. And we can expect 10 to the power of 10 to 11 transductants per day in the Mediterranean basin. So this means this mechanism should be extremely powerful to exchange genetic material between bacteria. But we still do not have very quantitative and precise data on that. So now we come to the viral mortality of prokaryotic plankton. So how do we estimate virus-mediated mortality? We can first estimate the bacterial cell lysis rate. And yesterday I talked to you about how we do viral production estimates, okay? If you divide this viral production by the burst size, and the burst size is the number of viruses released during cell lysis, then you get the bacterial cell lysis rate. And this can be expressed either as percentage of bacterial abundance that is lysed, for example, per day, or as percentage of bacterial cell production that is lysed. So that's one way to estimate this mortality rates, because that's what we need to understand how much cells can be removed by bacteria, by viral lysis of bacteria, and how this influences the biogeochemical cycles. And that's what I talked about yesterday also, is that we can estimate the frequency of infected cells. And then again, we can use that as a fraction of bacterial production lysed, or in a more sophisticated way, there have been models developed to make these estimates a bit more reliable. That has been done also with isolates. Okay, now I show you some data from the impact of viruses on the cycling of carbon in the deep sea sediments. On the right, you see all the different environments that were investigated. And the samples were taken in different water depths, but from the sediment, from the surface sediment, the first few centimeters. And the very interesting point here is that the virus-induced mortality increases with water depths. So we had a bit less than 20% virus-induced mortality in coastal waters. In mesopelagic depth, it was more than 50%, and almost the entire mortality of prokaryotes was due in the sediments, was due in the bathypelagic, the deep sea sediments, was due to viruses. And this should cause a cycling of dissolved organic matter by the viral shunt, and by that the losses of bacteria to the meiofauna, that is the nematodes, should be reduced, and therefore this is probably the reason why we found a formerly unexplained high bacterial activity in the deep sea. Now we go a bit to the food web dynamics. So what we are talking about here are the two predators and their prey. In blue you have the bacteria or the prokaryotes. In red you have the viruses, and in green that's a flagellate. Theoretically, you should expect that the flagellates and the viruses compete for prey, that is for bacteria. And this is just a sample from a lake, where you have here in the open squares is the viral lysis, and in the surface water, which is anoxic, the lysis was relatively low, about 20%, but in anoxic water, where there are not a lot of protists, viruses took over in terms of mortality. And for the grazing by the due to the flagellates, that the open circles here, there was an inverse trend found. You also see in this, what people also found is that in areas the salinity extremely high, like in high salinity lakes, viruses completely dominate the mortality. So this would indicate the competition between the two types of grazers. And lytic viruses can also be seen as predators. Okay, you have seen part of that yesterday. This just shows you how we do this kind of experiments. And here again is with 0.8 filtration, you can remove particles and protists. With the tangential flow filtration, you can concentrate the prokaryotes. With this ultrafiltration, you can make virus-free water, and that's new from compared to yesterday, you can make a virus concentrate. Then you can combine the prokaryote concentrate with virus-free water and with inactive viruses. You kill them in a microwave, or you don't kill them, so you have active viruses. You have a treatment with and a treatment without viruses. And then you can do experiments with that. In an extension of that, you can also do gravity filtration. That's like here 0.8, so you can concentrate the prokaryote, the flagellates as well. And then you can have 4 treatments, not only active and inactive viruses. And that would be like the control with no viruses or with plus viruses, but you can also have a treatment with flagellates and only prokaryotes and no viruses, or you have a fraction with viruses and with flagellates. And with that, you have 4 treatments, and you can investigate the relative effects of viruses and flagellates, or when they're both together, on the prokaryotic community. And most of the studies have been done here in freshwater system, because it's easier to do the concentration of viruses. And I show you a sample here. And on top in A and B, you see that the blue thing, that's a flagellate, and the green thing is a bacterium, a specific genus of bacterium that's called Lactobacillus, and be stained with a method that you have heard about probably already in this course, that's called FISH, or fluorescent in situ hybridization. And on the right side, you see that the flagellates can eat this bacterium because it's inside the cell. Treatment C, picture C and D, that's a micro-autoradiography, which means you add some radioactive material like thymidine to the samples, expose it a bit, and then also expose it to on a filter, to a film so this radioactivity makes silver grains. And 2 things you can see here is, in the presence of the flagellates only treatment without viruses, you get some filaments. You don't get that without the flagellates. And also you see that the black dots are the autoradiography grains, which means they take up this material. This is the thymidine. E and F is the treatment with flagellates and viruses, and you see that the filaments are much longer, and the silver grain load is much higher, which means the activity is even higher when both predators are present. So here we have a little table to summarize this data. So the abundance, so the second column is the flagellates, and the third one with flagellated viruses and see that in all of them, the abundance, the length of the whole filament, the length of single cells in a filament, and the percent of total bacterial volume, and the relative activity as grain area per filament is always much higher when flagellates and viruses were present compared to when only flagellates were present. When only viruses were present, there were no filaments. Okay? And these filaments are a protection form for these Lactobacillus against grazing by the flagellates, because these filaments cannot be taken up by the flagellates if they are getting too long. So, and the idea behind is why the viruses help, so to speak, the Lactobacillus, is that some bacterial populations are infected by the viruses, and so they release all this organic matter, these lysis products, and this can then be available for the Lactobacillus who cannot be eaten by the flagellates. So this shows a rather complex interaction between viruses and flagellates. There was also this theory by Miki and Yamamura that as protists are potentially graze, that they can graze on viruses and infected cells, and they should reduce infection. And they did that with a model, and they said, this model kills the killer of the Wiener hypothesis. So when doing this kind of experiment several times in freshwater, data show a completely different way. For example, on the left side, you see that the frequency of infected cells and the virus production in the virus-only treatment is much lower than in the viruses plus flagellate treatment. And similar data have also been found several times in different types of this environment. So obviously, viral lysis benefits from the presence of the flagellates. Oops. And that's also other studies have shown that. So this data seems to kill the killing of the killer of the Wiener hypothesis. And the reasons for that could be changes in diversity and therefore changes in infection. But it is also known that flagellate grazing, and I have shown you that in this viral shunt food web model, is that grazing releases inorganic and organic nutrients, and this stimulates the growth of bacteria, and stimulated growth of bacteria usually also means higher infection rates. There could also be inductions of lysogens, as I've talked about yesterday. This is a bit too complicated maybe, but these are a similar type of experiments from a marine system in Hong Kong, where the data show that the predators reduce the biomass accumulation, but they sustain diversity. You know, all the negative stuff here on the top is abundance, then production. But on the lower part, when viruses are present, there are always a stimulation of bacterial diversity. Let's skip that one. Okay, that's an experiment from the deep sea from 2,000 meters with and with viruses. And this data show a heat map and the relative abundance and activity of some operational taxonomic units. And the left heat map is DNA, and the right heat map is RNA. DNA usually roughly stands for the biomass part, and RNA stands for the more active part. And what you can see that without viruses, let's see on the top, in the dendrograms, without viruses, the samples are the most different from T0 or also different from the presence of viruses, which indicates that the presence of viruses also influences bacterial diversity in the deep sea. And that's another way to show that. But A and B show plus virus to the left, minus virus to the right with DNA. And you can clearly see that these networks analysis are different in colors and size. And the network analysis is nothing else than, in a way, than correlations, negative correlations here, blue, and the positive ones in red. And the lower part shows the same thing, but with RNA. So again, there's a strong difference between these samples. And that's for this experiment made with samples from 2,000 meters. Well, and that's, I think that's a bit too complicated but you can read that yourself if you want. But it clearly shows that I say that just in a sentence, that when you have the viruses removed, this decreases diversity and increases the connections, the positive connections, the positive correlations between some bacteria. And this would mean that some bacteria without viruses would take over the system. So the viruses prevent that, and this sustains a high diversity of the host bacteria. Okay, now a bit to the viral effects on carbon and nutrients. Again, the viral shunt and the food web scheme, but this time with some data. This data will depend on the system. This data will depend on the season, on water depths and everything. But this is just a summary that was made at that time. And this starts with the carbon flow through the system based on primary producers. That's set as 100%. Okay, that's what all the carbon that phytoplankton takes up with the aid of sunlight, and that's made of phytoplankton biomass. This carbon is set as 100%. And from that, between 6 and 26% is shunted by viral lysis of primary producers, grazers, carnivores, and heterotrophic prokaryotes into the dissolved organic matter pool. And that's quite a substantial number, indicating that viruses are very important in the cycling of organic matter. So, and that's done by lytic viruses and viral lysis. And so that's the release of the so-called lysis products, ribosomes, cell plasma, cell wall fragments, mitochondria. And this transforms autotrophic and heterotrophic cells into dissolved organic matter and cell debris. And this has been documented with so-called VHS, which means virus host systems. That's a virus isolated bacterium on the host, and also with natural communities. So what has been shown that polymeric DOM is produced, polymeric DOM in the sense of combined amino acids and cell wall compounds, but also in terms of colloids and empty cells. And studies have also shown, again with VHS, virus host system model communities and natural communities, that a significant part of these viral lysis products are very rapidly degraded and belong to the so-called LDOC pool, which means the labile DOC pool, which fuels then the bacteria and should increase the use of organic carbon by bacteria. And at the same time, then it's important, it also reduces the transfer to higher trophic levels. because cells that are lysed cannot be taken up anymore by prokaryotes, by protists. And this has also been demonstrated in a very elegant study of Middlebow, who added either only lysis products or virus host systems to a natural community. And what they found that bacterial production, respiration, enzymatic activity was going up, but another important point is that bacterial growth efficiency decreased. So this enhances bacterial production and respiration and reduces in bacterial growth efficiency by the viral lysis, reduces, as I said before already, the transfer of carbon to higher trophic levels. Okay. Well, that's second part is not important here. And these are data from different environments, MED1 and MED2 means the Mediterranean Sea. HNLC means high nutrient, low chlorophyll areas. That's in the Southern Ocean. And Bloom means it's also from the Southern Ocean, but where there was this area was fueled with iron, because this HNLC areas where it's not a lot of chlorophyll, but a lot of nutrients are limited by the availability of iron. But in all of this environment, when experiments were done, is bacterial respiration was stimulated in the presence of viruses, bacterial production was reduced, and bacterial growth efficiency was reduced. Bacterial growth efficiency is the amount of carbon that can be taken up by bacteria and is not put into respiration. And growth efficiency was roughly reduced by 35%. So the enhanced respiration, the enzymatic activity, and the reduced growth efficiency by the release of lysis products mean probably that by viral lysis more organic matter is processed, but turns into CO2 and not into biomass. And also that the greater percentage of the nutrients is remineralized because more dissolved organic matter is processed. And this would mean a lubrication of the microbial loop. And this is data from different environments, from the Mediterranean Sea, the Pacific, the Southern Ocean, which is a combination of real data and models. And this show that bacterial growth efficiency decreases with shunting efficiency. Shunting efficiency means here the stronger the impact of viral lysis, the higher, that's the shunting efficiency. The more higher the shunting efficiency, the more organic matter is transferred from the cells into the dissolved organic matter and can be used by viruses and by other bacteria. And it's very interesting that in this model and based on data, viral lysis was more important than temperature, because usually it's assumed that the higher the temperature is, the lower the growth efficiency. But obviously in this environment from Southern Ocean, where it's very cold, to the Northwestern Pacific, where it's rather warm, or the Mediterranean, this factor was more important than the temperature, than the classical explanation for bacterial growth efficiency. So what are the potential roles of viruses in the cycling of carbon and nutrients? Viral lysis converts cells and also cell production into dissolved phase. And this can result in an increased retention time of materials such as, or elements such as carbon, nitrogen and phosphorus in the euthotic zone, or iron also in the euthotic zone. And this can reduce the transfer of carbon to higher trophic levels. So this would be circle within the dissolved organic matter, bacteria, and virus part of the food web. And the increased bacterial respiration and reduced growth efficiency could also stimulate nutrient regeneration. And this data support the view that viral lysis acts, as Curtis Huttle called it, as catalysts in the nutrient sucking, and also that they lubricate the microbial loop. This is data from an in situ experiment, an iron fertilization experiment in the Southern Ocean, in the so-called high nutrient, low chlorophyll area. What's called here inside is the area where this, where a part of the ocean, a very small one, in an eddy was fertilized with iron, and outside, which means without iron. So you see all these parameters: viral production, frequency of infected cells, increase of in situ viral abundance was higher inside the iron fertilized patch than outside. And also here the data on release by viral lysis of iron and chlorophyll show the same kind of data. And here are data on rapid PCR. I've shown a little bit about that yesterday already. And you see here, circled in red, that also the diversity of the bacteria inside the patch then changed to another community compared when there was no iron fertilization. I think this is a bit difficult to read, but this was done during also in the Southern Ocean during a cruise, where you have the Kerguelen Islands. They are not easy to recognize, probably for you, but then you have different areas of chlorophyll, and what these islands do, again in these HNLC areas, these islands release iron from the sediments, so they produce phytoplankton blooms, and then studies were done outside and within these phytoplankton blooms to see how such a fertilization influences viral parameters and bacterial parameters. So here you have the iron fertilized stations versus the HNLC stations, high nutrient, low chlorophyll, and to very much to the right you see the ratio. You see that bacterial abundance, bacterial production, viral abundance, and viral production, as well as contact rates between viruses and bacteria. The amount of lyse bacteria and mortality was higher in these iron fertilized stations than when there was no nutrients. And this indicates also what we know from other studies is when bacteria grow well, they are also infected well. Okay, well, does this not continue? Okay, now. So from a biogeochemical base now perspective, there is still an ongoing debate whether viruses short-circuit the biological pump. I think you have heard about that already. If not, the biological pump is all this organic matter that is produced, for example, during decaying phytoplankton blooms, or by fecal pellets of zooplankton and material that is then sinking out of the surface ocean and transported into the deep sea, incorporated into the sediment. And this is very important because this removes carbon from the surface ocean. That's very important also for climate issues, because the carbon that is stored in the dissolved organic carbon, or as dissolved organic carbon in the ocean, is as high as all the carbon and the CO2 in the atmosphere and all the CO2 stored in animals or plants. Okay, so whatever happens to this biological pump is extremely crucial for biogeochemical cycles and also for climate. So viruses could short-circuit the biological pump just by lysing cells and removing lysing cells or particles by converting these particles, these cells, to dissolved organic matter. There's also another way, because viruses could also prime the biological pump in two ways, because infected cells sometimes start to sink. So this could increase the host import from the euphotic cell zone. Also, these lysis products, and I'll show you a little bit of data just in a minute, can increase the aggregation of organic matter, increase particle size, and this could also transfer carbon into the deep sea. So this release of sticky colloidal cellular components during lysis could be very important. Okay, so organic aggregates. I've shown that a little bit yesterday, but what do they mean for viruses? Are they vectors for viruses, which means they transport viruses? Are they scavengers of viruses and inactivate them, or are they factories of viruses? I show you some data now, but I give you right away a summary. The data suggests that aggregates are rather viral vectors and scavengers than viral factories in some environments, but not in all. So experiments have been done with where you put samples on so-called rolling clamps to the turn, so the material is not sinking out, and then you have some light. And in this experiment, you get usually a phytoplankton bloom and a lot of marine snow flocks. Okay, I have another slide before. To the left, you can check that yourself if you can other way to show the viral shunt, but also on the right to see the so-called viral shuttle. That means that viruses get trapped on organic material, for instance, produced by cell debris and cell fragments which aggregate and sink out. So they have the viral shunt would mean you convert cells into the dissolved phase, and the viral shuttle would mean that viruses get attached or produce even organic material, organic aggregates that sink down out of the environment. This is another way to look at ambient water and particles. So on both, you have in ambient water, you have a viral shunt, but also on the particles, you have a viral shunt. And what's also important here is that organic aggregates can float if they are buoyant, which means viruses can transport it along with currents to other environments, or they can sink out, but they can also rise again if there's enough chlorophyll in the particles, which produces gases like oxygen, and this is trapped in this organic matrix, and then these particles can also at some points rise again. This is from an experiment done in an environment, in a coral reef environment, with organic aggregates that were produced from diatoms artificially. And in this environment, it could clearly be shown that bacteria had a two-way motion. That is, they were absorbing and were released. So there were scavengers and factories for bacteria. But for viruses, they were mainly only the way to release viruses. So this would be viral factories. So that's also possible. Which shows you that also the different types of particles have to, in the future to be analyzed to see when organic aggregates are factories or scavengers or vectors. So this is the rolling tank experiment I was talking about before. And in red, this is just plain seawater. And in blue, there was a 2.5-fold enrichment with viruses. The bloom that developed was by diatoms, Chaetoceros, Costunidiscus, and Lichia. And the presence of viruses first caused a delay in the development of the bloom, but then the bloom was stronger than in the control. And one of the potential explanations is that the delay was caused by infection of diatoms, of course. And then the higher abundances of, or higher chlorophyll concentrations later on were caused by the viruses infecting phytoplankton and bacteria and acting as catalysts of nutrient cycling. That's not so important here. We also looked at, or this paper also looked at the size and stability of the marine slow flux. So ESD here means on the left side is particles with an equivalent spherical diameter of more than 1 millimeter. This was done by making pictures of that. And what you see clearly is that first you get very big particles also in the control, but these decay very fast. In the viral enrichment, these big particles persist much longer. And this could again result, for example, in a stronger biological pump. I'm going to go back to that again. And these larger particles, or the persistence of the larger particles, could be due to cell lysis products, like cell walls that are sticky, or colloids that are sticky, and that make these large marine snow particles more stable. All right. So prokaryotic viruses, of course, they are parasites of unicellular organisms. They are parasites, they're cell parasites. However, the ecological role of them is more complex. The lytic viruses, at least infecting single-celled organisms, are better described as predators. They have more to do with predators than with parasites. Viruses with a chronic infection, for example, that do not kill the cell but release a virus once in a while, they are really true parasites. And some temporary viruses or lysogenic interactions where the genome of the viruses is kept within the host genome, these are probably better described as mutualisms. And from a viriocentric point of view, the host cells are the main resource for the viruses, and they serve as phage factories or phage bioreactors. So while grazers have bacteria as resource, viruses have cells the same way as resource, but it's really a resource for them. In a very similar way as the grazers, they take everything from them, including the metabolism, because viruses do not have a metabolism. Okay, now towards the end, I show you some phage oddities of this, from the strange world of the viruses. And that's here an example of the Lavida viruses. And that's the case of the giant viruses, or they're also called gyruses, and the virophages. What you see here on the left in blue, it's a giant virus that's infecting an amoeba. And in red you have a virus that co-infects with this large virus. Sputnik doesn't mean the vaccine here, okay? And what happens, and the virophage is an incomplete phage. But when the giant virus infects the host cell, it also replicates the DNA that is necessary for the virophage. So the virophage infects the giant viruses within the cell to reproduce itself. So viruses can infect other viruses. We also know that a lot of single-celled organisms try to shut off their metabolism when they're infected, apoptosis that's called. But for example, for the cyanophage genome, we know that they encode for a photosynthesis gene, which means when the cyanobacteria that are infected want to shut down the metabolism, the virus forces the metabolism to sustain photosynthesis in the infected host, which results in increased viral production. So the cyanophages stole the photosynthesis system too from the hosts. There are also other genes that are proper viral inventions that you do not find in cells. For example, the cholera toxin that you find in the Vibrio cholera, which causes cholera, is not a bacterial gene. It sits on a prophage within the host genome, but it sits in the virus. And Vibrio cholera is always a nasty bacterium, but if there's no prophage in cells of Vibrio cholera, you don't get cholera. So this is a viral invention. Another one is a specific repair system that's called the gene DAN5 that has never been found in the host genome. It's only found in bacteria, in bacteriophages and also in algal viruses. And it makes, for instance, the algal virus chlorella almost impossible to get killed by UV light. Another phage oddity are experiments where people try to study the effect of viruses on Synechococcus, but what they found is like the approach I showed you yesterday, by removing the viruses, or today also, by removing the viruses and then incubating, and you see the number of viruses coming out, and looking at the growth of the host. But what was found here is when you had the viruses inactivated, and that's the open bars, the closed bars, cyanobacteria didn't grow anymore, or didn't grow very well. And this is probably due to, again, to this recycling or catalyst capacity of nutrient cycling of heterotrophic bacteria in the presence of viruses. Okay, so cyanobacteria, Synechococcus, needs viruses to infect heterotrophic bacteria to grow well. That's another story from, I think, salt lakes in Tanzania. You know, they have in these salt lakes, there are a lot of flamingos, and flamingos eat only cyanobacteria. They are toxic to all other animals or birds, but not for the flamingos. And in this in situ survey, it could be shown that in situations where viruses were controlling the abundance of cyanobacteria, they were indirectly also controlling the abundance of flamingos because they were controlling the food. So the impact of viruses shows in this very specific ecosystem an enormous bottom-up trophic cascade for the flamingos. Well, you can wonder yourself whether this is a spaceship landing on a planet or a virus landing on a cell. So climate change, I have just one slide for that, because we don't only know very little about how viruses are involved in climate change. Climate change, if it's a change in temperature, for example, in the water column, you will get changes in the growth rates of bacteria or the host cells, and this will change the infections. There are also lots of speculations that there are outbreaks of viral infections, but the point is, currently, and that's why I'm not talking much more about that, there's a lot of ideas what could happen, but we have not a lot of hard data that could make us build model of what could happen. Okay, and this is the end of the talk. And I hope I can go back to you to answer some questions. So what I do now. I don't know how to, I do not hear, if you want to ask questions, either write me or because I don't know how to, go back to to speak to you. Chat, is that maybe that? Oh, I should have asked Begonia how to do that.
Yes, I have a question, Professor.
Okay, I hear you.
Yeah, because I was like raising my hand in the chat, but you weren't able to see it. In this diagram of the viral shunt that you showed us, you said that up to 26% of viruses conform the dissolved organic matter when they are dealt with by grazers, if I understand the diagram correctly.
What I'm saying is that if you set the phytoplankton carbon production to 100%,
mm-hmm.
then up to 26% of this carbon is entering the dissolved organic carbon or dissolved organic matter pool by viral lysis or by viral lysis products.
Okay, thank you.
Mm-hmm.
Hello.
Hello?
This is Melina. Yeah, I have a question. I don't know if you could go back to the slide 19.
Let me see. 19. What's that?
It's an example of the PLOVC about lysis and grazing lifestyles and bacterial mortality. Because it says like grazing dominated in the epithelium and lysis.
Epithelium, that's the euphotic layer, yeah.
Okay, and hypolimnium?
That's the anoxic layer.
Okay. Okay, thank you. Yeah, I was confused about the terms.
Yeah, well, that's from a freshwater system. So the epithelium would be the euphotic zone. And then you have, I don't know why I come back to that. Yeah, somehow the system got stuck. Yes, but the point is in the anoxic layer, that's in the deeper lake, there are almost no grazers, but viruses can take the mortality rates. And in surface water, at least for this lake, grazing was more important than lysis. That's the point I wanted to make. Does that answer your question?
Yeah, yeah. Thank you, thank you.
Okay, any other questions? Okay. If there are no further questions, I think we can stop the course, terminate the course. As I said yesterday, if something is unclear, you have my email address and you can always write to me if you want. Okay, here I get something written. From Juana. No, this is a misunderstanding. It's the mortality rates of viruses in the bacteria due to viruses in the deep sea are not necessarily higher than in the surface ocean. It's a misunderstanding. And anyway, what one has to consider here is, it's not a bad question, it also depends, mortality is always given in a relative way and not in an absolute way. You're of course right, the majority of the production occurs in the ocean, in the euphotic ocean, but you still can have in one system, like the surface system, for example, or let's say in a coastal system, where the production is even higher than in an offshore system, you can have 10% mortality due to viruses, and in the offshore system you have 50% of mortality due to viruses. But in absolute terms, you will still have higher mortality if you look at production cell lysis rate in coastal systems. But usually we give mortality as percentage of the production, so in a relative way. Yes, right. What you're writing is okay. All right. When nothing else comes in, I wish you a good continuation of your course. And again, if you have questions, please do not hesitate to contact me. Goodbye.